giving the recording. Right. Okay, boys. Once again, good morning. And we are into an interesting chapter. As I said, molecular inheritance. And in this chapter, basically, we discuss all the molecules that are involved in our body, particularly in the genetic material. Already you know that our genetic material is in the form of two nucleic acids. One is DNA, another is RNA. DNA, deoxyribonucleic acid, RNA, ribulose nucleic acid, or normally we say ribose nucleic acid. And both these DNA as well as RNA are present in the chromosomes. And all of you know, chromosomes in turn are present in the nucleus of the cell. So basically, when you search for genetic material, you know that you need to search in the nucleus because that is the location. So basically, you have a cell, a living cell, and in the living cell there is a fluid, liquid present, cytoplasm. In the cytoplasm, as we discussed, many cell organelles will be there. And in that cell organelles, one of the cell organelles, very prominent one, is nucleus. So you have a living cell, inside that nucleus. Inside the nucleus, another liquid is there, it is called nucleoplasm. Inside that nucleoplasm, there will be chromosomes, ribbon-like structures, thread-like structures, and inside those chromosomes, the genetic material will be present, DNA and RNA. And inside the DNA, there will be what we call genes. And genes are responsible for our characters. Already in genetics chapter, we talked a lot about varieties of genes. So basically, the location of genetic material is inside the DNA. This, thanks to the technology that we have developed, we understand easily now. But way back in 1928, a gentleman called Frederick Griffith, he got the idea, first idea, where is it located, our genetic material, and is it transferable from one generation to another generation. So, many questions were circulated in the world, and to answer for those questions, many scientists across the globe conducted experiments. And we should appreciate all those gentlemen, because even gentlewomen, because technology was not there. It was basically their presumptions and assumptions. And that is the reason why. Yeah, I am coming to that. So, Frederick Griffith conducted an experiment which is known as Griffith's transforming principle experiment. Okay, the name of this experiment is Griffith's transforming principle experiment. Now look at the gentleman's photo. So we need to salute him for the work he has done. And when he named it as transforming, means something present in one cell is moving across to another cell, from one person to another person, from one generation to another generation. Okay, and there is a principle involved. It is not that straight it goes. There are certain guidelines which need to be followed. So what are those principles involved in the transformation, and what is being transformed? That is the basic question that Griffith wanted to answer. So what he has done, he has taken two organisms for his experiment. One is mice, another is bacteria called Streptococcus pneumoniae. You know, coccus variety it is. That's why the name Streptococcus. And the species name is pneumoniae because it causes a disease called pneumonia. And you know, pneumonia is a lung disorder. There is accumulation of water in the lungs. So lung infection basically. So in that Griffith identified two strains. Two strains means two varieties. One is the S strain S stands for smooth strain. What is this smooth strain? Why is it so smooth? It has polysaccharide mucus coat, and you can see that orange color coat around the polysaccharides. Okay, and that makes the surface smooth in these bacteria. That's why we call the S strain. And it is virulent. Virulent means highly infective. It causes infections. That is virulent. The second variety is R strain, rough strain. In this, no mucus coat will be there in the polysaccharides. Now look at that. The blue colored one. Since there is no outer coat, mucus coat, the surface of these polysaccharides will be rough. Hence, R strain. So, in the bacteria, based on the presence and absence of mucus coat, there are two types of strains. Smooth strain called S strain, okay, and it is virulent because it causes the disease pneumonia. And the second one is rough strain because the surface of the polysaccharides are rough. There is no mucus coat. But this doesn't cause pneumonia. Non-virulent. I hope it is clear. Have you understood? And of course, basically you are all chemistry students. Saccharide means sugar. Isn't it? So polysaccharide. Many sugar units are joined together. It's a polymer. So to that polymer polysaccharide, there is an outer coat in the form of mucus coat. Presence of mucus coat around the polysaccharide makes it smooth. Hence, bacteria having such polysaccharides is referred to as smooth strain, or in short, S strain. Hope it's clear? And the other one, having no mucus coat, whose surface is rough, is labeled as R strain. Out of the two, who is dangerous? Yeah, S strain is dangerous because it causes pneumonia. That's why we are calling it as virulent. And R strain is non-virulent. It doesn't cause pneumonia. That is the difference. Right. What is that he has done? Frederick Griffith. Look at that. Now he has taken a beaker, glass beaker, right? Inside that he has taken living S strain bacteria. Remember, basically when we say S strain, you must remember now onwards it is bacteria, Streptococcus pneumoniae. And he has taken a needle, a syringe, taken that live S strain with the medium, injected into the mice. Mice means small rat, small sized rat is called mice. And what happened? The moment he injected the S strain bacteria into mice, what happened? Mice died. So what does it mean? Why the
It is harmful.
the mice died because S strain has the potential to cause pneumonia disease to rat.
Pneumonia.
Yeah. And just now I said pneumonia is lung disease. The moment it is injected, the lungs of the mice must have been affected and breathing problem would have been there. Due to lack of breathing and respiration, the mice died. So only we call S strain as virulent, something that causes disease. So first step in the experiment that he has performed is he took the S strain with the media, injected into the mice, and mice died. So he has come to a conclusion that there is something in the S strain that transformed into the mice and killed the mice. Okay? He only said there is something. He did not tell DNA, right? Because I remind you that there was no technology at that time. 1928 experiment, right? Second step, he has taken live R strain. Again, R strain, rough strain bacteria, injected into the mice. Of course, another mice, right? And the mice continue to be alive. That means there's something good in the R strain which has gone into the mice but did not kill the mice, allowed the mice to be live. That means he has confirmed with these two steps that S strain is a dangerous fellow, R strain is a good bacteria. Okay? Now, he did not stop the experiment there. He took the S strain, third step, step number 3. Okay? And he boiled it. So, heat killed. When he boiled, what happened? S strain died. Correct? Because of boiling. After all, it is bacteria. The moment he boiled, S strain is killed. So, we say heat killed. And that heat killed bacteria he injected into another mice. And what happened? Surprisingly, mice is alive. So the mouse leaves no bacteria cells found in the heart. Now, step number 4, what is that he has done? See, a lot of thinking. That's why I always tell Sandeep, Kadkol, Prasad, Rakesh Mohanty, think out of hat. Whenever we are having some problem, normal thinking will not help us. You need to think out of the box, isn't it? And those who think out of box, you will clear your SSB, isn't it? And I have been telling you, situation reaction test, SRT. During your SRT, you need to think out of box, because in your batch, 8 people will be there. So the test is common for all of you. Whoever thinks out of box will be the winner. If you remember, when you were in 11th class, I was telling you in the class, whether you do any physical exercise or not, whether Rakesh is doing PT or not, whether Kadkol is swimming or not, whether Prasad is doing horse riding or not. What is important? Thinking, thinking and thinking. Wherever you are, whatever you are doing, think. Even a farmer, when he is plowing the land, he thinks a lot. He thinks of the coming rains, he thinks of the coming monsoon, he thinks of the minerals present in the soil, isn't it? Even though he may not be educated, but he is having culture. That is the thing. So this man also thankfully thought, and as I said, out of box thinking. What he has done in step number 4, he has taken the heat killed S strain plus mixed that with live R strain. Heat killed S strain and live R strain mixer. Rakesh calls it a chow bath. Okay? And injected into the mice. What happened, Rakesh?
Mice died.
Ram Naam Satya hai. Mouse died. Mouse died. Now, like Sandeep, he also got doubt. Why? Kyun? Kya ho gaya? Why mouse died? Okay? Yes. Then lot of thinking. How is that Rakesh, a good boy, becoming dash? Because of whom? Isn't it? Here, who is the good boy? R strain. Isn't it? But why he is becoming bad? Because of friendship. With whom friendship? Yes, strain. And Kadkol always tells in the mic on the stage, habits die hard. You see, even though this fellow S strain is heat killed, its nasty habit is there still. What is that nasty habit? Killing the people, killing the person, killing the rat, killing the mouse. So that killer instinct present in S strain is transformed into good bacteria, R strain. And R strain, because of what Prasad calls Savasa dosha, bad friendship. So R strain also became bad boy and killed the mouse. So Griffith said that R strain, which was non-virulent, became virulent and killed the mouse because of its friendship. So step number 4, S strain heat killed plus R strain which is live injected into the mouse, and Rakesh, mice died. Ram naam sach hai ho gaya. Hence, what is the conclusion? Now look at that. Griffith concluded that, and he was very intelligent. Griffith concluded that some transforming principle. See, he did not tell DNA, RNA, gene, nothing, because he was helpless that time, 1928. Some transforming principle transferred from heat-killed S strain to R strain. Isn't it? Something from heat-killed S strain transferred into R strain, and R strain became virulent. And that enabled R strain, okay, to synthesize, what is that? Smooth polysaccharide coat and become virulent. So change in the structure of sugars has made this bacteria either virulent or non-virulent. Rough polysaccharide is non-virulent. Smooth polysaccharide is virulent. So unfortunately, R strain, which will be having normally rough surface of the polysaccharide, because of its dosti, friendship, became smooth, and that made the R strain to become virulent. So this is due to what is said, transfer of some genetic material. Of course, now Rakesh knows what is that genetic material, why R strain becoming bad. Why? Who is after all giving order for the cell to prepare a saccharide, a sugar molecule? For that matter, any molecule in our body, who is the command and control? Our Prasad calls it CO, commanding officer. Who is the commanding officer for a cell? It's the nucleus. It's the nucleus that gives order. And inside the nucleus, just now I said, chromosomes. Inside the chromosomes, DNA. Portion of DNA, gene. Portion of DNA is gene. Last chapter, previous session we studied structure of DNA. Remember that helix, double helix model? 3.4 angstrom units I said? Yes, that is it. So, Griffith's experiment concluded that there is something that is making changes. Transforming principle. Now, with the help of technology, we know that the transforming principle is nothing but DNA. But before we know, already 3 scientists, again, our salutes to these 3 scientists. Let me show you the photo of these 3 gentlemen. Look at that. Again, thinking out of hat, Kadkol. These 3 people. And it is related to some of your favorite subject, biochemistry. I know somebody like our Mahesh is interested in biology as well as chemistry. So that combination, Mahesh, biochemistry. Biochemical characterization of transforming. The moment Griffith announced to the world this experiment and results of that experiment, naturally people started thinking, what is the transforming principle that made R strain, which was good bacteria, became bad bacteria? How is that? Presence of a smooth coat or rough coat in the polysaccharide would make a bacteria bad fellow or good fellow. And who is giving order for this? So they wanted to answer such questions. And that is the reason why I was always asking you, ask questions. Ask questions. Ask questions to
Sir,
your... Yes, please.
Sir, when the S train is heated, sir, why that transforming
Yes. When S strain is heated...
it should also means when S train is heated, sir, transforming principle should also be killed, no, sir?
Correct. but it did not kill, it got transferred. Means with the heat that is supplied, we understand that that transforming principle could not be killed, it was hidden. Rest of the things, cytoplasm, other things got dead. That is the thing.
Yes, sir.
Isn't it? You just think of it.
Yes, sir.
How is that only that transforming principle is alive in spite of heating? That means it has got something. And now after this experiment you will understand, Niranjan, that these three scientists answered your question. What is that?
Yes, sir.
One is
DNS.
why DNA? Let us understand. Okay. Basically, I will let us first salute these scientists. One is Oswald Avery, another is Colin McLeod, and third one is Maclean McCarthy. So these three together work to determine the biochemical nature of transforming principle. Like Niranjan, they too got doubt. Okay, first thing, question number one is, what is the transforming principle? Because Griffith did not tell the DNA is the transforming principle. He said something is there. Some transforming principle is there in S type, S strain bacteria, and that got transferred. But he did not name it. Now these three scientists wanted to find out what is the transforming principle. What are the chemicals that are present in that? What is the behavior of that? And who is giving order for that to transform from one cell to another cell? Okay? Now a number of questions they wanted to answer. And what they have done, they started guessing again. Lot many experiments, Kadkol, will go by guessing. And our Sindhu knows that we need to first have a plan before we embark on a journey. What our Sindhu will do? He will plan where to take the vehicle, where to stop for lunch, how to carry breakfast, isn't it? Like that, they thought that yes, maybe proteins must be the reason, maybe DNA, maybe RNA, like that. So, whenever I ask you, last week you wrote MCQ, isn't it, Niranjan?
Yes, sir.
In that, let us say, Niranjan did not understand one question. Then what he will do? But he has to attempt. He has to take a chance because negative mark is not there. If it is right, luckily you get mark. If it is wrong, 0, no problem. But at least you have satisfaction that you tried. So what Niranjan will do? He will apply logic and he will remove possible wrong answers one by one. Okay? Deletion of possible wrong answer. And what is left out, like A, B, C, D, first Niranjan will feel D is not at all related to question. So D is out. C, probability is there. So he keeps a doubt. B, no way connected. It is the same chapter but topic is different. B is out. D is out. Now the choice for Niranjan is between A and C. Now he will start thinking, analyzing between the two alternatives and arrives at a conclusion that A must be probably. He is not sure because he has not studied. It is, unexpected question. That is the reason why I tell always Kadkol, expect the unexpected. I tell Sandeep, prepare for the unexpected one so that you will not be disappointed. Right? So here also what they have done, possibly they had 3 alternatives. You are having 4 alternatives in your MCQ. These fellows have got 3. One is protein, second is DNA, third is RNA. So what they have done, luckily by then some time passed and they got what is called enzymes. They have come to know. So they added enzymes. And enzymes are the biochemical substances that act or dissolve. Isn't it? Like digestive enzymes. Whatever we eat, digestive enzymes digest the food so that you are getting energy. So they used first enzymes called proteases. When they used that enzyme protease, they ensured that all proteins are dissolved and conducted that Griffith experiment and found that still transforming principle is taking place. That means it is not proteins. Next, they took another enzyme called RNase. And with the help of RNase, RNA is dissolved. But still, when Griffith's experiment is repeated, the transforming principle is taking place. That means they understood it is not RNA which is involved. Now they took another enzyme, DNase. Remember I told you in the class, enzyme's name will end up with, yes, Kadkol? Yeah, ASE. Remember in the class, 11th class, biochemical molecules, I said sucrose, OSE, maltose, OSE, glucose, OSE, ending with OSE is sweet boy like Kadkol, sugar molecule. But if you do spelling mistake, Rakesh, OSE replaced with ASE, sucrase, maltase, glucose, you add ASE in place of OSE, that becomes enzyme. So here proteinase, RNase, DNase, it gives a clue to Prasad that an enzyme that dissolves the particular thing. Proteases dissolve proteins, RNase dissolves RNA, DNase dissolves DNA. So the moment the DNA is added, okay, what happened to Prasad? DNA is digested and transforming principle did not take place. See, so nice experiment they conducted. First they ensured proteins are digested, conducted Griffith's experiment, transforming principle took, and the mouse died. That means the proteins are not responsible for the death of mouse. Then they conducted RNase. RNA dissolved, conducted the experiment, still the mouse died. Now DNA is dissolved. When DNA is dissolved, the rat is alive. That means DNA is responsible for the transforming principle. That means DNA has given the order for the polysaccharides to have that smooth coat. Because in the experiment that we described just now, what makes
Yes, strain is heat killed, sir.
It is heat killed.
DNA does not get digested.
Yes, exactly. We only see what they have done, Sandeep, only they boiled. They did not use enzyme, no. Perhaps Griffith was not knowing what, was not having the knowledge of enzymes by then. Understood? See, most of the experiments during that period, it is only physical exercise, physical experiments. Now that we have got everything, including the electron microscope, nanotechnology we have got now. Now we can prove any experiment, isn't it? Anything.
Sir,
Yes.
sir, the transforming principle is transformed from S strain to R strain or R strain to S strain, sir?
I said good boy is becoming bad boy. Good boy, who is good boy here? R strain.
R strain.
Yes. He is becoming bad boy. Who is bad boy? S strain. Okay? That means transforming principle, in fact, even S strain, Niranjan, is virulent or bad because DNA present in that is giving order for the polysaccharides to have smooth coat. Isn't it? So this became bad. Anything that has got a smooth coat of polysaccharide is a virulent. So just for our understanding,
S strain is
heat killed. Yes, Niranjan, go ahead.
So when S strain is heat killed, sir, when it is mixed with R strain, transforming principle will go from R strain to S strain, sir.
It is S strain to R strain. Because who is bad in that? Out of the two, S or R? It is
yes,
S strain. S strain, correct? S strain is having DNA which is making smooth polysaccharides. presence of smooth polysaccharides will make disease to the organism to which it is in, like in this case, mouse. Correct now?
Yes, sir.
So what he thought, Griffith, when he boiled the S strain, he thought everything will be dead. But he did not realize that DNA will be still alive in spite of boiling. So that is the transforming principle. So DNA from S strain, even though it is heat killed, transferred to RNA. And I mean rough, not RNA, rough strain, R strain. So R strain also became unfortunately virulent. Understood? To ascertain that, they have used enzymes, these three gentlemen. Now look at that. What is that they discovered? Let us conclude before we run out of time. Digestion of protein and RNA. Just now I said, how to digest protein? By using an enzyme called protease. See the spelling? ASE. And RNase did not affect the transformation. So transforming substance was neither protein nor RNA. But digestion of DNA with DNase inhibited transformation. It means DNA caused transformation of R cells to S cells. Isn't it? So there is a change. The moment DNA from S strain gone to R cells became S cells. Hence, that is the reason why I said yesterday, your parents will be always asking you, who is your friend? Isn't it, Kadkol? Always they will be inquiring about your friends. And some psychologist said that if Kadkol names 5 friends of his, I can know the personality of Kadkol. Oh, even though Kadkol is R type, how many S types are there surrounding Kadkol? Isn't it, Kadkol? Yes, Niranjan, understood? Yeah, let me show the experiment that they have done in the form of a diagram. Look at that. Let me expand. Are you able to see?
Yes, sir.
Yeah. So first it is heat killed S cells. First lipids are removed, carbohydrates are removed. And how to remove these lipids and carbohydrates? Sandeep? You know that sedimentation process, heaviness. And then what is left out? Lipids are out, carbohydrates are out. By just using some chemicals, I am sure in your chemistry lab you must be removing them. Like Sandeep must be using again an enzyme, fat dissolving enzymes. So fats are out. Lipases. Remember, Sandeep? Use lipase, lipids are out. And sugars, just now I said, glucose, maltose, glucase, maltase, sucrase, ASE. The moment you start using this, these are out. So once in the heat killed S cells, lipids are out, carbohydrates are out. What is left out, Niranjan? Only 3: proteins, RNA, DNA. Correct? Now,
Sir, protein or proteases, sir?
protein is the chemical substance, food substance. Protease, Rakesh, is enzyme. Okay? Both are chemical substances,
Yes.
but they differ in function. Protein makes Rakesh He-Man. Okay?
Yes, sir. Yes.
Rakesh becomes Mr. Orissa, Mr. Odia, because of proteins. And when Rakesh eats a lot of proteins, what is making the proteins to get dissolved? You see, protein is a polymer, as all of you know. And what is the monomer, Rakesh? Amino acids.
Sir?
Amino acids.
Yes, sir.
Amino acids join together to make a protein. So when proteins are broken, amino acids are formed, Rakesh is building his muscles. He becomes Mr. Odia. Right? Is it not Kadkol? Sandeep, of late you are not seen Rakesh. He is building a lot of muscles. I am seeing him in the campus. Yes, Kadkol? Make one video call to Rakesh, you will see. What do you say, Rakesh? Okay? So, proteins will be dissolved, Rakesh, by using enzymes called proteases. And after dissolving proteins, what these three scientists have done? They added R cells, rough strain, and no transforming. Right? Still, transformation occurs. That means the S cells remain. From S cells to R cells, transforming principle has been taking place. Next, they dissolved RNA by using RNase, and still transformation is occurring. That means R cells are becoming S cells. Correct, Niranjan?
Yes, sir.
Yeah, I am happy Niranjan has understood. Now the third one is, left out one is DNA. And DNA gets dissolved by using an enzyme called DNase. And when DNA...
has no role.
RNA has no role. That is what they have proved, no? Out of 5, see, in any living organism, 5 things will be there, Sandeep. Fats, carbohydrates, proteins, RNA, DNA. The question that poses in front of us is, who is this transforming principle out of the 5? So thanks to these 3 gentlemen, McCarthy and McLeod, they proved DNA is the transforming principle. Why? When DNA is dissolved in them, the R cells, so look at that, remained as R. No S cells found. So that means no transformation. So if DNA is not there, no transformation. That means we have ruled out one by one. The transforming principle is not fats by dissolving fats. The transforming principle is not carbohydrates by dissolving carbohydrates. Transforming principle is not proteins by dissolving proteins. Transforming principle is not, Sandeep, RNA. And what is transforming principle? It is the DNA. How do you know? R cells, okay, remained R cells. Like our Sandeep, good boy, remained as good boy. It did not become bad boy. When R cells remained R cells, what happened? No transformation. Understood, Niranjan? I hope all of you understood.
Yes, sir. Understood.
Yeah, very good. So tomorrow I will take up one more because it is showing time, right? Hershey and Chase experiment. Tomorrow we will discuss. Take care. Bye.